drugs, or ideas, what you consume influences who you become. On the Mind and Matter podcast, we learn together from the best scientists and thinkers alive today about how your mind-body reacts to what you feed it. Before starting Mind and Matter, I spent 10 years in academia doing scientific research. I got a PhD in neuroscience where I focused on neuroendocrinology and the neurobiology of behavior, and before that, I specialized in molecular, developmental, and evolutionary genetics. I use my scientific background to help parse and translate the information that guests share on the podcast. In addition to the podcast, I write long-form written content inspired by the show where I integrate what I've learned across episodes. I also have a free weekly newsletter where I provide you with upcoming guests, share links, and provide commentary on scientific studies and research that I'm reading, and more. Visit mindandmatter.substack.com to find all of my content. You are about to listen to the full episode, but there will be 1 to 2 short ads immediately following this before the conversation. If you would like to hear ad-free episodes and get early access before they release more widely, become a paid subscriber at the Mind and Matter Substack at mindandmatter.substack.com. Becoming a paid subscriber is a way to directly support my efforts, allowing me to make more episodes and higher quality episodes, and also helping to free up my time so that I can spend more of it on doing research and synthesizing results from the podcast and from the literature so that I can do more things like writing on the Substack, putting more content into the newsletter, and getting these things out more frequently. So if you want to support the show and get ad-free episodes, go to mindandmatter.substack.com. Hey everyone, I want to take just a minute to tell you about a really cool health monitoring device I've been using called Lumen. It's a handheld, pocket-sized device with a sleek design. The Lumen device measures CO2 levels in your breath, which allows their technology to determine the extent to which your body is burning fats versus carbohydrates. Lumen helps improve your metabolic flexibility, your body's efficiency in shifting between using fats and carbs for energy. I use this device in the morning or before bed or after meals or after workouts to track my metabolism. With just a couple weeks of use, I learned a lot about which foods were causing my body to burn mostly fat, mostly carbs, or both, as well as how long I needed to fast and how hard I needed to exercise to promote fat burning. Being able to track whether your body is burning fat versus carbs will help you figure out how your body responds to your lifestyle and whether or not your weight loss and fitness strategies are actually working. Lumen is great for anyone looking to optimize their health for either weight loss or athletic performance. Their easy-to-use app allows you to track your results together with what you're eating and how you're exercising, and it syncs with other devices like the Apple Watch. Click the link in the episode description to learn more and use the code MINDMATTER, MINDMATTER, to get 10% off your Lumen device today. That's on top of the site-wide sale they are currently running, and the Lumen device is eligible to be paid for with FSA and HSA health accounts. Check out the link in the episode description for more info. Thank you for joining me.
Thank you.
You want to just start off by telling everyone a little bit about who you are and what your lab studies?
Okay, I'm a professor of environmental health sciences at the Mailman School of Public Health at Columbia University. I also have an appointment in molecular pharmacology and therapeutics in the School of College and Physicians here at Columbia. And I'm the director of the Center for Innovative Exposomics at Columbia. So my background is in pharmacology and toxicology and neuroscience. And I'm just very interested in how environmental factors contribute to human disease.
Mm-hmm. And you've done a lot of work that has to do with neurodegenerative diseases like Parkinson's and Alzheimer's, right?
Yes.
Can you give us just a basic overview on neurodegeneration here? So like when we think about Parkinson's and Alzheimer's in particular, or neurodegenerative diseases in general, compared to other types of brain issues, psychiatric issues, what are neurodegenerative diseases as a class? What really characterizes them?
So when you think about the major ones like Parkinson's and Alzheimer's, to me, one of the most critical features is how age is such a major driver of it. So when you look at people that are over 65 or 70, then the percentage of people that will develop one of those two diseases continues to increase. And so if you think about where we were 100 years ago when the lifespan was much shorter, you didn't see as much of it because we weren't living to these older ages. But now, through all the advances in modern medicine and the lifespan being up in the high 70s, you're now getting to this point where a large percentage of people will develop these diseases later in life. In general, they're characterized by, it's a progressive disease. They get worse with age and they continue to get worse. And early on in the diagnosis, the symptoms are relatively minor, but then they progress. And so when you think about like from a treatment standpoint, while cures would be nice, slowing the progression could really increase the quality of life because it takes like decades for these things to really come together. And so I think it's thinking about how you can intervene to slow the progression is a very important thing to think about. It's not always about like preventing it totally, which would be good, but the idea is if you can slow the progression, that would be a big advance. I think people, if you know people that have Alzheimer's disease, you go from like forgetfulness to not being able to recognize somebody. And there's a big range in between there. And so the idea is that can we slow that progress so that someone dies of other conditions when they're 90 and they never get to that point of like totally losing their memory and ability to recall people.
Yeah, and I would imagine that the earlier you start, the bigger the dividends. Like even if you only slow something a little bit, if you start early on, because these things can take years to play out and to fully manifest in some of these symptoms, and eventually for the cells to die, even if you do what you might think is a small boost or help to the problem early on, that might give you years of extra time.
All right. Oh, what's really amazing, like in Parkinson's disease, is that the symptoms that lead someone to go to the doctor don't tend to occur until the majority of the neurons in these key areas in the substantia nigra die. So the idea is you can lose half of those dopamine neurons and have no symptoms. So if you could slow that down, you might get to the point of, like, so if you're in a high risk category and you have a genetic risk factor, that if you could slow it down, you may never even get diagnosed with it because there's that much reserve in there by the time someone actually has the physical symptoms.
Yeah. And so I guess then the question becomes, if people typically aren't going in and getting diagnosed until they're relatively advanced in some of these conditions, half or more of their neurons have already died, the dopamine neurons for Parkinson's. The question then for me is, okay, how do you get people to detect things before those symptoms arise? Are there milder symptoms that we just don't recognize as symptoms, or are there sort of preventive tests that can be done before there's any symptoms?
Yeah, so I'm not a physician, but I've worked with many of them, and I have a pretty decent understanding of the disease. Like in Parkinson's disease, the resting tremor is a very common feature. But another big part of it is kind of like shuffled gait and how people walk. You can detect this quite early. There's also things like with arm swing. And there was a really interesting presentation I've seen some other people many years ago where after Michael J. Fox was diagnosed with Parkinson's, someone went back and because he's on TV or movies for his entire life and went back to see when they were seeing symptoms way before he knew he had it. And the idea is that you go back in time and you can see this. Another interesting symptom is micrographia. People's handwriting starts getting smaller. And so there's these really good, back when we used to do accounting by hand, of people that developed it where they saw the handwriting had changed a decade or more before they even knew they had the disease. And so when there's these subtle sort of things that are there, you can imagine something like with how we interact with a phone or a computer, that they should be able to pick up these at a more subtle level earlier on that may allow us to diagnose them sooner. So again, it's something where with all of the new devices we have, and if, like when you think about like shuffled gait and walking slower, can a Apple Watch or a Ring detect those sorts of things? And I think that's where we're starting to see that potential that these minor things could be picked up much earlier with this more advanced sort of technology we have now.
Yeah, yeah. And not only earlier, but in a non-invasive way, because, I mean, if I'm hearing you right, you're basically saying there are relatively subtle motor signals that one sees relatively early in some of these neurodegenerative diseases. And they're signal there. You can see them if you know how to look for them, but they're subtle enough that people probably don't notice them unless they have this in mind already. And, given that we all have mega computers in our pockets now with video capabilities, it's easy to imagine, especially with AI getting better every day, that you could upload, say, a time series of videos of yourself and actually potentially see some signs here of early neurodegeneration.
Yeah, and I think one of the challenges is the specificity is that there's other reasons you might have some of those symptoms, and so I think this is like where the AI becomes very important. Is people with Parkinson's also lose their sense of smell.
Hmm.
There's many other reasons people lose their sense of smell, but it does happen. And so the combination of like, are you having motor symptoms? Have you lost your sense of smell? Are you having GI problems? And I think it's going to take that sort of feeding in for information from multiple levels. But I agree. I think with having the devices that we have around us and things, I really think that there's potential to detect these things much sooner. And I think from a therapeutic standpoint, we've tested many drugs for many different diseases, and I think so many of the failures have come because they were started too late. So we might have drugs in the arsenal that if you could start them a decade earlier could work,
Mhm.
but you have to have that consistent diagnosis earlier.
Yeah, yeah. And because I would imagine that because these diseases take years and years to play out, going from subtle symptoms to medium symptoms to devastating symptoms, that could be decades of time. So if you could buy someone a few years by starting earlier, that could really make a difference.
Right, absolutely. And that's the idea is that we, even for something like Parkinson's right now, the main treatment, which is replacement of the dopamine using L-DOPA, uh, it works really well for 5 or 6 or 7 years, and then people start getting really bad side effects. And so if you could have someone be able to wait 5 or 6 more years before they started that could, again, really increase the quality of life by just pushing it off a few more years.
Hmm. And so that's essentially what Parkinson's is, right? I think you said this briefly earlier. You're losing particular neurons in particular parts of the brain that release dopamine. And so one of the ways to treat this is just to give someone exogenous dopamine to counteract the degeneration of the dopamine neurons that are in their brain. But you said that only works temporarily.
Yeah, I mean, it's something where you start getting more and more side effects as the disease progresses more.
Hmm.
And so you may have like this 5 or 6 or 7 year honeymoon where the L-DOPA is working really well. And I think the other thing that, like, while we know it's always been defined as this disease of having a low dopamine, which was discovered back in the 60s, it's not just a disease of dopamine.
Hmm.
To me, it's a classic defining symptom of it. But there's many other things going on in the body. It's like when I mentioned the GI things, people develop constipation, they lose their sense of smell. They often have like hypotension sort of issues. It's a whole body thing, like this alpha synuclein protein building up in different areas. And I think this is like one of these places where there's other opportunities for earlier diagnosis because it's not just these dopamine neurons that are located centrally in your brain that we can't biopsy, but there's things that you can actually see in your gut way before as well too. And so I think this is like where looking at it as a multi-system disease, but there's no doubt it converges at this when the dopamine is lost. That's when you get those hallmark tremors, slow movement, and things that really are kind of the defining part of it. But
Hmm.
it starts earlier. A great example is that many years ago, it was some Scandinavian country where they were cutting the vagus nerve coming from the gut up to the central nervous system as a treatment for ulcers. And the people that had that had a much lower incidence of Parkinson's. And it started pointing to people that the sort of gut brain connection was a key feature in it, and that there was some potentially movement of like the synuclein, the pathogenic versions of synuclein were kind of causing that. So again, it's a whole body thing, but it like all roads lead to that loss of dopamine.
Hmm. Hmm. So to what extent, so when we think about a neurodegenerative disease like Parkinson's, to what extent is this just a general degeneration process? I'm getting older, neurons in my brain are starting to die all over the place, but I just happen to lose these dopamine neurons relatively early and it manifests as Parkinson's, versus it being a specific degeneration of those neurons in that part of my brain.
Yeah, I mean, so as we age, we lose a lot of function in many different systems. The thinking with the dopamine neurons is the molecule of dopamine is very reactive. And if you like take some dopamine and put it in a test tube and leave it on the bench, it'll turn black. It'll oxidize and turn black.
Hmm.
And so being a reactive molecule, those neurons that have a reactive molecule in them they're just like living in this system that is very reactive. And so the thinking is that they are a vulnerable population. So if something happens that makes you not be able to handle that as well, that would make it worse. And so my lab has spent a lot of time studying how the dopamine is packaged inside the cell. So the idea is the cytosol, if the dopamine is not in the vesicle where it's normally stored and it leaks out into the cytosol, it's a much more reactive environment. Mm-hmm. So the idea is if it's packaged in the little vesicles, which it tends to do in recycling, it's safer back in there. But if you, for example, if the protein that packages dopamine into the vesicles is called VMAT2, when we knock that out in a mouse, those mice develop a Parkinson's condition because they can't keep the dopamine in the right place. They get the reactive intermediates and those dopamine neurons die. So the idea is that the dopamine neurons are vulnerable neurons. And I think if everyone lived to be 115 or something, we would all get Parkinson's eventually. So it's a vulnerable population, but there's very clearly genetic and environmental factors that accelerate the process. And so the people that get diagnosed are the ones that have that acceleration of that degeneration of those neurons.
Mm-hmm. So these neurons are inherently vulnerable because dopamine itself is reactive. It is easy to oxidize this molecule. And because we breathe oxygen, because our metabolism inherently uses oxygen and we create reactive oxygen species, if the cell isn't careful to protect and package that reactive dopamine molecule in safe places, in vesicles, it's naturally going to get oxidized, and that's just going to cause problems.
Exactly. And a good example of this from like other conditions is that there's pretty strong evidence that people that take amphetamine type compounds for extended period of time, whether it be methamphetamine as a drug abuser or amphetamines for treating things like ADHD, it increases the likelihood of someone getting Parkinson's disease. And these amphetamine compounds release dopamine from those storage vesicles. And so It makes sense in this idea is if you disrupt that storage, you are increasing the likelihood of that oxidation occurring.
I see. So in general, even if this is somewhat simplistic, for whatever reason, the more dopamine that gets out of those vesicles, the higher the odds that it's going to be in this that's going to become oxidized outside of the protective environment of the vesicle.
Exactly.
And is this how in Parkinson's, is this how these dopamine neurons actually die? It's the oxidation of dopamine specifically that's involved in that?
It's a contributing force to it. Mm-hmm. So again, like when you have like synuclein, the pathogenic protein, it's in many different neurons in the brain. But there's data from a paper from Peter Lansbury's lab many years ago that showed that the reactive forms of dopamine stabilized the dangerous forms of synuclein. Like, so the idea is that even though synuclein can be in many different places, when it's near that oxidized dopamine, it's even worse.
Yeah.
So again, it's not like exclusively these dopamine neurons that are dying, but that's like where it's the worst. And so it's one of the contributing factors. And it's, to me, that reactive environment in the dopamine neurons make them more vulnerable, but it's not the only pathway to having the damage to the neurons.
Mm-hmm. So based on the things you've told me so far, I want to ask you this question. Obviously, neurodegenerative diseases like Parkinson's will become more common over time just because, in general, human beings are living longer. But if we control for age, is Parkinson's becoming more common? Are more people in their 60s getting it today than got it, a few decades ago?
So, I mean, it's a very exacting epidemiological question. And from the data that I've been looking at over the past 20 or 30 years, I'm not convinced that there's an overall major increase. I think it's mainly this increase in age, but also increased awareness and diagnosis. Like it's
Hmm.
very hard to tease those things out. So you might see an increased number over time, but the way we detect it and diagnose it has also gotten better.
Right, right.
So I'm not convinced that we see a wholesale increase, but when you look in certain populations, there definitely seem to be times where there's occupational sort of things or things where people do have a higher incidence. And so I think it's the increased aging population is the biggest driving force. Whether or not there's other things, I'm not as convinced of that.
Mm-hmm, mm-hmm. So everyone is more prone to neurodegeneration with age. For Parkinson's in particular, other than age, what are any other risk factors that we know about, whether it's dietary components or environmental chemical exposures, genetic factors, anything else besides age itself?
So head trauma is a risk factor, and much of that is thought to the initiation of an ongoing inflammatory cascade that just continues. So that's what the head trauma seems to be from that. And it seems like if you have, this is like documented concussion, loss of consciousness level sort of measurements. There's many environmental factors that have been implicated. Pesticide exposure is a risk factor, which is something that got me first interested in this. But there's hundreds and hundreds of pesticides out there. And so like pinpointing it has been difficult. The other thing that my lab has been extremely interested in are some of the things that decrease the likelihood. So caffeine consumption and primarily cigarette smoking appear to decrease the incidence of it. And so there's still environmental factors, but like what's in those things that are causing it? Like those could be insights and ways of potentially decreasing the progression.
So caffeine consumption and cigarette smoking actually predict lower incidence of Parkinson's. I've actually heard both of these things, and I wanted to ask you about the latter and if we knew anything more specific about it. But that is in fact an observation that's true.
Yeah. So the caffeine one is very well studied, and I think that because we have so much data on caffeine and coffee and tea consumption, and even where people drink decaffeinated beverages, like so you can kind of tease out that part of it. So that one's been pretty strong, and the adenosine signaling seems to be a big part of it. The tobacco and the cigarette smoking has been harder to get at because tobacco smoke is very complicated, thousands of molecules in it. And so a paper from Haida Payami at University of Alabama, Birmingham, probably about 12 years ago, really shifted a lot of my research. So she was doing a genome-wide association study to look at drivers of Parkinson's. But besides just looking at whether or not you get it, she was aware of this effect of smoking. And she's like, well, what if we did a GWAS to see if something was modifying whether or not cigarette smoking was protective? And in that study, she identified a protein called the synaptic vesicle glycoprotein 2C, SV2C. And then they said, well, this could this is just humans. It's smoking, very complicated thing. And so she did work in Drosophila. So went to a fly model, treated them with a toxicant, and then used nicotine to see if she could protect from it.
Pure
and
nicotine.
then redid the GWAS in flies and pulled up the ortholog to SV2C.
So that term ortholog, what you're basically saying here is we're talking about a protein that is part of the synaptic vesicles that importantly store the dopamine that we were talking about before. And I think what you're getting at here is that this must be a highly conserved protein that has a similar core function in vesicles in flies and other animals, including humans.
Yes. And when I saw that, like to me, when you look at the environmental factors in Parkinson's, the smoking protection is actually stronger than like the pesticide making it worse. And it's like this really jumped out to me. And so when I heard about this data, and I knew one of the co-authors, I called up Haida right away and was like probing her for information. So my lab went and knocked out SV2C in mice. When that paper came out, there were only like 8 references to SV2C in the literature.
Wow. So it's this core protein, highly conserved, but it was not well
totally understudied. Yeah. A member of the family, SV2A, is the target of Keppra or levetiracetam, which is like the biggest selling anti-epilepsy drug. So SV2A was really well known, but SV2C, no one knew anything about it. So we knock it out in mice. and then we measure how well they release dopamine using voltammetry. And when you did that, they release less dopamine.
And that would predict protection against Parkinson's.
Well, they released less. They had less dopamine, so they were actually lower functioning. Like the idea is when you didn't have SV2C, you couldn't store dopamine as well into the vesicle.
Okay, so it did not store as well in the vesicles and therefore got oxidized more, presumably, and that would be a problem.
But it was really nice kind of confirming her results. And now since then, SV2C has shown up just in regular GWAS for Parkinson's as being an independent genetic risk factor for it. But it lives on that vesicle, which my lab has been studying for years. And so my lab has been working on SV2C ever since then.
Yeah. And so obviously there's a huge bag of worms here because we're talking about nicotine. But with respect to this result, nicotine appears to have this anti-Parkinsonian effect. Is it fair to say that's established?
Yeah, so and there's nicotinic receptors on the dopamine neurons, and there's many different pathways involved with nicotine. But another study from some colleagues, about Alberto Serio at Harvard, did a study on nutrition, looking at people that had diets high in food that were high in nicotinoids. And they had a lower incidence of Parkinson's.
So nicotinoids would just be any compound that can activate a nicotinic acetylcholine receptor?
Yeah, a lot of peppers.
Okay.
So the idea is that it wasn't like a lot of people at first were saying, is it something about smokers and addictive personality sort of thing? But the idea that nicotinic foods could also have the protection, made us think that it very much was the nicotine and not something else in the cigarettes.
So the fly study, the GWAS study in flies that you mentioned, specifically used nicotine. So there's one piece of evidence. Exactly. And now this other piece of evidence is this more general observation that nicotinoids, foods with nicotine-like compounds, have similar pharmacology. They have, repeat the observation one more time. They have lower incidence of Parkinson's?
Wait, the mice...
The dietary thing that you just mentioned,
the people that had diets high in the nicotinoids had a lower incidence of Parkinson's. Okay. So the more nicotine you take in, it appears to be protective to Parkinson's disease.
Wow, interesting. And then caffeine as well has some kind of effect?
Yeah, so caffeine seems to do a similar effect. Now, caffeine and coffee consumption has been shown to have positive health associations for many different things. So I have no problem encouraging people to drink coffee and tea. We've been doing it for thousands of years. But cigarette smoking, we know, increases the incidence of about 100 other diseases. So the idea of cigarette smoking being there seems something that you can't do. But the idea that, that activating that system, one of my initial sort of very simplistic ideas of this is like the nicotine exercising the dopamine neurons? Like, is it making them fire more? Like the idea is that, like we know from other studies that people that start getting Parkinson's disease, they start moving less. And that's being sedentary makes everything worse. The idea is you need to keep these neurons going. And so the idea is that physical activity is something you always want someone who's in the early stages because it seems to help that. So is the nicotine kind of artificially activating and exercising the neurons is one of the ideas that might be why it has that protective effect.
So, okay, wow. And then you said amphetamines basically correlate with higher incidence of Parkinson's. And, I don't know how deeply you want to go into that, but obviously a big question there is, we are giving people a lot of amphetamine prescriptions these days, starting at a very early age. Is there anything worth getting into there about, say, Adderall prescription frequency and Parkinson's?
So, I think with my view of most medications is that you should take the least amount you need for the effect you're trying to get. People that have ADHD and are unmedicated are at higher risks of many bad things, higher levels of incarceration. There's a lot of bad things that happen. So being medicated for ADHD usually has a positive life effect. And even though you may slightly increase your incidence of getting Parkinson's when you're 80, you have a better chance of probably getting to 80, right? So it's one of these things like where the idea of like indiscriminate use of ADHD medications in kids, that does concern me because we have other non-amphetamine drugs that can treat it.
Hmm.
And I would always want to look at those first to see if they work. But if you have a person who's taking those medications, like, are they taking it every day of the week, or are they taking it the days they're going to school? Or, it's like you want
Yeah.
to decrease the amount you take. But I don't like, I'm not against people taking those medications because they do have a major effect in improving quality of life. And so it's like you're trying to balance those things out, but you can't ignore that it is increasing the risk of this condition that's linked to the dopamine system.
Hmm. What kinds of studies have been done linking amphetamines to Parkinson's? Because, one point of context I want to give to people here is nobody's done a study where you take all the kids who've been on Adderall for 25 years and compared rates of Parkinson's to the kids who haven't, just because that study is never going to get done. I would imagine the links here come from studies that occur with much shorter durations.
So there's a few studies on this. Again, the ones with like people that are using methamphetamine, like when you're looking at that's a different thing when people are drug abusers, but they have an increased incidence.
Yeah.
But we've been using these stimulant medications for decades.
Hmm.
And so these are situations where people have taken these things. Again, I don't know what the actual age groups and things with this, but it's data from thousands of people that were taking amphetamines. Maybe they were pilots or something. We used to prescribe these things in the military and stuff. Like,
Yeah.
so it
Yeah.
can go back decades. And so it was people that had like, multiple years of regular usage. I don't recall how far back they went, but it's not a big step to say, well, if you're starting these when you're 7 or 8, my personal view is that giving medications to a child that's 5 or 6 because they can't sit still, that doesn't make much sense. There's many other things we can do before the medications come in. But when the medications, when the behavioral things can't work, and it's kind of like, okay, we've tried these other things, we're working on that, and you want to bring the medication in, like that's the sort of progression you want to do, because getting that child to concentrate and not get in trouble and get arrested or something, that's like a very important life outcome from that. So again, it just gets back to the idea of using these drugs judiciously and recognizing that there are long-term risks of long-term use of it.
Mm-hmm. Mm-hmm. So the genome is the full set of genes that one has. The proteome is the full set of proteins one has. You've studied something called the exposome. Let's just give people a crash course. What is the exposome, and how did you get into this?
Yeah, so 20 years ago, Chris Wilde, who served as the director of the International Agency for Research on Cancer at the World Health Organization, he was a cancer epidemiologist, and the Human Genome Project was being completed. He saw this great success from that. But he also knew that cancers had an environmental role. And he's like, so now we have this whole complement of the genome. Like, what if we had the entire set of our exposures throughout our life? And he said, it'd be great to be able to complement the genome with the exposome. So he wrote a commentary on that, saying this would be a great thing to do. And then for 5 years, there wasn't another paper on the topic. And then there was a meeting at the National Academy of Sciences, and Steve Rappaport and Martin Smith kind of pushed this sort of idea, like, maybe we can try to do this. And so they held this meeting in 2010, I believe. And being at the National Academy of Sciences, it got a lot of attention. One of my friends went to the meeting. He came back from it. And he said, oh, have you heard about this? And I'm like, no, but it sounds pretty interesting. And so we invited a lot of the speakers at that meeting I was at Emory University at the time, just to come in a symposium to talk about what this is and could we do it. And I think one of the first things I recognized was that it was a great concept, but we couldn't measure it yet. But it wasn't like we couldn't do it. It was like we just have to figure out how to do it.
Mm-hmm.
And so what connected this back to what, like my earlier mention about pesticides and Parkinson's disease, I got very frustrated trying to study a disease where there's hundreds and hundreds of pesticides, and we studied them one at a time. It was like
Yeah.
whack-a-mole trying to do that. I'm like, if I could study those at an exposome level, like that sounds great. And so what Dean Jones and I at Emory said is like, well, why don't we start building the infrastructure for that? So we wrote this center grant to develop the platform for measuring the exposome. We weren't ready to do it. We were like, let's build the tools for it. And so we applied for this grant to NIH, and it got rejected as being too crazy. Then we ended up getting it a year later. And so we started developing the tools for this. And so Dean was working on high resolution mass spectrometry. These instruments had been getting better and better each year. And we knew that the data would be very critical. So we built up like a data sciences and systems biology core. And just really started working on how you could measure as much as possible. Now, people have been very good about measuring things in our environment. Like we can measure with monitoring systems and different devices. But what Dean and I saw as being very critical was that we know that individual variability is very critical. You can have people in the same household have very different outcomes. Twin studies show people have very different outcomes. So what we thought was best was like, well, what if we just focus on measuring things in people, like in their blood?
Mm-hmm.
So let's start measuring as many of these environmental chemicals as we can, but at the same time measuring the endogenous biology. So in metabolomics, we measure all the small molecules we can, all the things in Krebs cycle and glycolysis. You're measuring all what you have in your biochemistry. But most people in metabolomics were ignoring the exogenous molecules. If they saw a plasticizer, they would think it would be an artifact. If they saw a pet, they didn't care about that.
Mm-hmm.
It wasn't in the biochemistry textbook, so they were ignoring it. We're like, why don't we try to get better at measuring those environmental chemicals? And so really that was what like the next 7 or 8 years was just, we were just doing method development. We were just trying to get better and better at measuring these things. And getting to the point where it was actually something that we could actually do. And I think that we're there now. We can now do studies of thousands of people and use high resolution mass spectrometry. We use liquid chromatography and gas chromatography to get really good coverage of chemical space. And we just try to measure as much as we can in people's blood.
Okay, so you basically you take blood samples from people and you're measuring as much as you can. On the one side, we would like as scientists to know all of the exogenous, all of the foreign stuff that gets into someone's body, everything that they're exposed to. That's the exposome. The other side of this is basically what's all of the endogenous stuff, all of the metabolites that are supposed to be there, so to speak. That's essentially the metabolomics side. What you're saying is there's actually added value to having both simultaneously, because you can start to ask questions like, okay, well, if two people are exposed to one exogenous chemical, why does one person respond poorly and one person doesn't? Maybe they have an endogenous substance that counteracts it or something like that.
Exactly. And so this is like where I really have been a fan of like network science, because like I used to think about like biochemistry as pathways, but they're really networks, right? These things are all interconnected. And so the idea is that because we're measuring the exogenous and endogenous at the same time, you can see like as certain chemicals go up, what else is changing on the endogenous biology, and you're doing it on the same sample.
Right.
It's in that same drop you're doing it on. And I think that's what really helps us tie the exposure part to the biology. And so then if you start thinking about this at a multi-omic standpoint, if someone has data on the proteome or the epigenome or the genome, you can start saying, well, you know, what parts of it are genetically conferred and which ones might be coming from these exposures. And you can do this in sort of a multi-omic framework. So to me, it is something about trying to get as much information as you can, like in that biological sample. But at the same time, if you see this pattern of exposure in a population or in a person, can we kind of like back calculate where the exposure may have come from? So like, are there patterns of exposure for someone who's exposed to diesel exhaust or to, you know, whatever the sort of occupational thing would be? So we think it's really critical to think about how we connect those air monitoring stations and the devices on, you know, and sensors to what's happening in the biology. So that's a key part. Is we're not saying it's all going to be done by measuring the blood, But when you combine that sort of geospatial data with the endogenous thing, that's when you really start piecing these things together.
Mhm. When you do exposome type studies, you measure all of the stuff someone has been exposed to in their blood. Are there any particular chemicals people get exposed to that pop up more when you look at people, say, with Parkinson's disease or Alzheimer's disease?
Yeah. So, a lot of these studies are underway. There's many, we're doing these right now. But again, this idea that pesticide exposure being a risk factor, this is like something we've wanted to get at for a long time. And people have done studies where they will measure like a panel of pesticides, but they're only measuring what they're looking for, right? It's very much
Yeah.
looking under the street lamp. So just recently, some of my colleagues in Europe, Roel Mullion and Susan Peters, they were able to get samples from a study called EPIC, where and I'm thinking about here in this thing because we haven't published all this, but I'll give general sort of findings from it. Was the people entered the study back about 25 years ago, and they were following them for cancer, actually.
Hmm.
But hundreds of them developed Parkinson's disease because it was a study of like 500,000 people. It was a huge
Yeah.
study. And so we were able to get blood samples from when they entered the study. And
Hmm.
it was about 8 years on average when they were diagnosed from Parkinson's later.
Hmm.
So we had the blood samples before they even knew they had the disease.
Yeah. So you got years of data.
Yes. And
Wow.
so we said, well, let's look at the blood. And so the first thing we did is kind of like a general check was that they knew people had self-reported data on how much coffee they drank. European study, different countries, espresso, different things they did, but they do know their coffee. They could describe it well. And so we said, well, what if we just first like look at all the caffeine metabolites in these people in this snapshot of time 25 years ago. And so we had quintiles of how much coffee they reported they drank.
So despite the limitation of self-report data, it's subjective. You're drinking espresso, I'm drinking weak drip coffee. You can actually measure directly in all of those samples, the metabolites of caffeine.
Exactly. So we're measuring caffeine, theophylline, paraxanthine. And I think this is like a key thing when we think about looking at exposures is that it's not just that parent molecule. You want to look at all the metabolites. So we can measure like a dozen metabolites of caffeine. And the self-reported consumption like matched up perfectly with the caffeine metabolites we measured. Now, and we also, as expected, the people that had the highest levels of caffeine had the lowest incidence of Parkinson's because it's protective. And so it gave us like this quick check that what that self-report was matched up. And I think when you think about self-report data, is there's some things people self-report well and some things they don't. Pack years of smoking is quite accurate. And apparently caffeine consumption from their coffee is pretty accurate too.
Yeah.
But dietary factors are not as accurate. But so what we did was that we are looking at all of the peaks on the mass spectrometer. We're not saying we're going to look just at these pesticides. We're looking at all of the peaks. But we're able to identify hundreds of pesticides in these samples. And so we were able to go through and essentially do a pesticide-wide association study. I mean, it was really like an environmental-wide association study, but we had hundreds of pesticides in the analysis. And so by doing that, we could then see like of all these different pesticides, which of these were elevated 8 years before they were diagnosed. Now, does that give you causation? Can't say that one caused that, but people's exposures, while they do vary day to day, people tend to live in the same community and have similar occupations over time. And so even if you can only get a snapshot in time, it still tells a lot about what's happening there. And so we're able to do this. We're working on the paper right now. But like we did find some compounds that hadn't previously been connected to it, but there's no confirmation bias here. We were just looking at whatever molecules were there. But clearly we need to go back into the laboratory and show, do these chemicals damage certain features? And like in an animal model, we see it, but it allows us to do that complement to that genome-wide association study for a particular disease.
Yeah. So in a study like this, the advantage is you can do things in an unbiased fashion. You don't go in thinking we're going to look for this molecule and this molecule because those are the only ones we know about. You can detect sort of anything.
Exactly.
Yeah.
And even the detection, like a lot of the peaks that we see may not be identifiable. Like we know
Yeah,
exactly what the mass is, but they may not be in a database. And so, like what I like to say, we have a lot of unknowns, but they're not unknowable.
right.
It's just a matter, like you got to spend time figuring out what they are, but there's ways of determining what they are. But the idea is if you see this peak always showing up in
Yeah,
a certain
yeah,
condition, then you put that postdoc on it and say, figure out what it
yeah.
is.
Yeah, before you do a study like this, there's lots of unknown unknowns. Then after you do it, you've got a lot of known unknowns.
Exactly.
Yeah. Interesting. Okay, and obviously, again, these things you're not proving what does or does not cause Parkinson's, but these are very good clues that can then be you can then use to go in and do functional tests in the lab in an animal, say.
And then also, if you have genetic data, you can do things like Mendelian randomization on those human studies. I mean, so there's a lot more you can do, but we just have not historically had this systematic and comprehensive analysis of all of these exogenous compounds.
Mm-hmm. Mm-hmm. Um, so when you say the exposome, in my reading of your work, you're talking about the lifetime everything that someone is exposed to over the course of their whole life. How do you even start to think about how you would track that in humans? Are you doing studies where you're like these are going to be multi-year long studies and you're taking samples at regular intervals?
So this was something that it seemed daunting when I first heard of this concept. What my approach to this was that if a person goes in to see their doctor because they're ill, the doctor might take a blood sample from them and run a sample on them. So it's like you are who you are the day you're being seen by the doctor. And it's a result of all of your past history. So even if I don't have blood samples from the previous 50 years, what can I measure today? So let's say, for example, you were exposed to some very terrible chemical when you were 8 years old, and it caused these epigenetic changes in you that are changing your biology. There should be evidence of that changed biology when you're at the doctor's office. So you may not see the chemical itself that caused it,
Mm-hmm.
but you might be seeing the disrupted biology that was caused by
Yeah,
it.
you can see a
So,
biological echo of a previous exposure.
yeah, and I think this is like where we have a lot of work to do to kind of piece these things together. But there's a lot of studies that have been conducted where they had samples from people at multiple time points and they're biobanked. And so the idea is that can we go back in and take advantage of those sort of studies to really kind of piece together that life history and also use things like epigenetics and protein modifications and even some of the geospatial data about where they lived and worked and piece those things together. So the sort of life course piece of it is a complicated puzzle that will be hard to pull all those things together. But I don't think you always need to have that entire thing because if we can show that a person with a certain exposure profile won't respond as well to a drug or will have a faster progression of a disease, that can be actionable that day. And so it's like, yes, I'd love to have an annual visit and a blood sample from everybody to piece these things together. We can still work with what we have at this point in time. But we do want to get the sort of life course effect of it. It's just that I think snapshots can still tell you quite a bit.
Hmm. How do you start to think about studying things like exposures in utero or just an exposure that a woman has while she's pregnant? Part of the reason I bring this up is, these are for both the mother and the developing baby, these are sensitive periods of development. And just to give one point of contact here, I had a podcast that was really interesting with a man named Angel Nadal, and he studies bisphenol A, BPA, microplastics, and other endocrine disruptors. And one of the interesting things he shared with me is these things will disrupt endocrine signaling in anyone. But, if you were to expose someone to some amount of BPA as a healthy adult, it'll basically be some temporary messing with insulin signaling, say. But if you give that same exposure to, say, a pregnant woman, because she's in the sensitive period where her body is rewiring itself to accommodate the baby, those endocrine disrupted effects can get locked into place and stay with her permanently even after the pregnancy ends. And so given that this is such a sensitive period for a pregnant woman or for the developing child, how do we start to think about testing the effects of chemical exposures during those periods of life? Is that something where we really have to focus on animal models, or have you done any work there in humans as well?
So my own lab hasn't done this sort of work, but I have colleagues that do this, is that There's many studies where they follow pregnant women and they take blood samples from them throughout, through the different trimesters, and they can get cord blood, they can get placental samples, and then follow the child afterwards. And I think those are the types of studies that start getting you those data points. So by measuring the mother's blood sample, you get an idea of their exposures.
Mm-hmm.
And then, like you said, if there's changes in biology, changes in endocrine pathways, those are things that can be measured in the offspring over time. And so I know many people that are working on these sorts of studies, and they're getting that sort of data and getting that cord blood, the placental samples. And so it's a, to me, it is a something you can study this in humans. Like it works, like you can measure these things. And it's just a matter of the right types of samples and like what are those critical windows in there. Again, some animal studies are needed to really figure out some of those parts of it, but those human studies are underway, and I think we're seeing that. We know that high levels of exposures lead to worse, outcomes for the offspring. But pinpointing which ones are the worst, like that's the sort of work we need to do now that we have these tools to look at not just the one chemical, but all of chemical space in those critical periods.
Yeah, and so on the one hand, when we do, there's one type of screen you can do where you sort of know what you're looking for ahead of time, and you have reason to believe, say, a chemical is going to cause some effect. You can go in and look for it. But what you've been talking about is you guys are doing these unbiased screens, so you can kind of catch anything that's there, even if we don't even know what it is yet. Has anything, let's just talk about a particular neurodegenerative disease, I guess. When we think about something like Alzheimer's, are there any particular chemical exposures that we know seem to matter for Alzheimer's disease progression or risk?
So I have multiple studies underway now, where the sort of thing, like what we did, for example, in Parkinson's, we're doing them. I don't have the results yet.
Mm-hmm.
I think there is some evidence that exposure to some organochlorine type things like the DDT type compounds, we've seen things like elevated DDE in some of the populations. And so when you think about, these are compounds we banned in the 70s, but we can still measure in most people. They're very persistent compounds. And so if you think about people that are now in their 70s or 80s when those exposures occurred, we do think that some of the very toxic sort of pesticides may contribute to the disease. But for people that are now like 50 or 60, those exposures have been going down. And so
I see.
This is like where I know like the National Institute of Aging has recently funded multiple studies in this space. I don't think we have a really good answer, but I think the answers are coming. And I think that in some ways that you're looking at things where people have exposures that it could be a combination of the chemical or the sort of life that led to that exposure. So if you're exposed to a lot of environmental and industrial chemicals, you're going to have an increased risk of many diseases. And so it's like, how do you start teasing some of those things apart? So to me, like I'm still kind of like holding out on the Alzheimer's side because there haven't been as many things that have jumped out. Whereas like Parkinson's had a lot of other evidence of not just the pesticides. There's actually very large studies of solvents like trichloroethylene have been shown to increase the risk of the disease. On the Alzheimer's side, the main thing we've really seen is air pollution.
Mm-hmm.
And again, when we say air pollution, PM 2.5, well, what is that? There's a lot of things in there, but there's definitely good evidence that the air pollution is worse. But like, why is that? Because if you have increased risk of stroke, which could occur from the air pollution, that increases your risk of Alzheimer's. So is it a direct thing to the Alzheimer's or not? But I think that's where the studies that we have, where we can combine air pollution monitoring with the blood samples, like those are the types of things that we can start addressing. And again, I think these studies are underway, and I think we've seen some of these things with the air pollution that seem to link up. And I hopefully, like in a year, I'll be able to tell you a lot more.
Yeah. And, one way it might be natural for people to think about this or to hope things work is, that you find one chemical and that one causes Parkinson's, and you find one chemical and that one causes Alzheimer's. And to me, the danger there is that you run into the same trap that people and whole fields ran into in the early genome sequencing days, where we were hoping to find one gene for this and one gene for that. But to tie this back to what you're talking about with Parkinson's and the basic biology underlying that, if a lot of these neurodegenerative problems come from the oxidation of molecules, and oxidative metabolism is a very general thing. There's a lot of ways for a lot of chemicals to generate reactive oxygen species and to cause that type of thing. I would imagine that part of what we're going to discover in this whole area is, there isn't just going to be one chemical for this neurodegenerative disease and one chemical for that one, but many chemicals in combination just increase your risk to many of these things.
Right. I think we're going to see a lot of that sort of convergence. The idea is that, if you go back to the Industrial Revolution, like the exposures we had, 150 years ago were so high. And so we know that we need to reduce lead. We've reduced lead dramatically in many situations. But we have like over 80,000 chemicals in commerce now. And so I think it is going to be these sort of multiple combined exposures that are increasing these risks. Very much like how we look at like polygenic risk scores. Like the idea is you're going to find these sort of like polyexposome or polyexposure scores and it's going to be that exposure profile that shows the increased risk, not the exposure just to one or two chemicals. It's like, what is that combination or profile that an individual has?
Mm-hmm. Mm-hmm. When we think about the exposome, things that we're exposed to, if we take that in the most general terms, one big piece of that are exogenous synthetic compounds. But of course, there's lots of stuff that we're exposed to that we put into our body on purpose that is supposed to be there in some sense. And that includes the things that we eat, the things that we consider to be food. Is there anything interesting or notable that you're working on that has to do with exposome as it relates to dietary components?
Yeah, I think there's a few ways of thinking about our diet. Like we think about the nutrients we bring into our body,
Mm-hmm.
it's like that you need to run things, but all the things we eat are also carriers of other things.
Mm.
And this, like a lot of things we've seen, like in ultra processed foods and the sort of very clear indications of adverse health outcomes from it, is you have to kind of separate the nutrient from what else is in there. And so these ultra processed foods have many other chemicals and things that come along with them. And I think this is a key part of what we can do with exposomics is that, is your exposure to PFAS or some plasticizer, is it coming from where you work or from your house or apartment, or is it coming into your diet? It doesn't really matter. We're going to measure all those things in a person.
Mm-hmm.
And I think this is like where, to me, like one of the bigger concerns is how complicated our foods have become in the sort of ultra processed way, where the more things are processed, the more of those chemicals and additives we have in those foods.
Mm-hmm. Mm-hmm.
And I think that they've been understudied. And this is an area where it's not going to be just that one sort of silver bullet thing. You're going to see it's a combination of multiple compounds that are involved with that. So the more processed your foods are, the more plasticizers are going to come in with it.
Mm-hmm.
More PFAS that's going to come in with it. And so, the idea of like having more like our sort of explosion of fast food in our diet, it's not good. The more things you do to foods, the more chemicals you're starting to introduce to it. So it is something where, the recommendation of like, making your own meals and things and using fresh vegetables and ingredients has stood the test of time of being better for you.
Yeah, yeah. And I think all you're really saying there is, when we say things get added to foods, it's not just that, and I mean, this does happen sometimes. Sometimes, a food processor will add chemicals on purpose, and sometimes they'll induce chemical transformations on purpose. But these things can also be added passively, right? Just the more times you touch packaging made out of plastic or go through a machine that has some metal or another in it, you're just going to naturally pick these things up more and more, right?
Right. So often like these things, like it's not on the ingredient list,
right,
but it's in the packaging.
Mm-hmm.
And like, yeah, so the idea is if you microwave something in plastic like that, those things leach into the food you're doing. And so it wasn't made by the person who made the food.
Mm-hmm.
It was in that sort of processing step. And the other thing too, like a lot of the things we add in, into our food are preservatives. And I like preservatives. I don't want mold growing on my food. And I've told students before is that if you've ever seen mold on your food, just think back to the day before when you didn't see it,
Hmm.
it was there.
Yeah.
You couldn't see it, but it was there. And so, some things we want to preserve because we don't want to have molds and things. Like some of the worst things we have are these natural toxins that grow on molds and things like that. So there's a lot of natural things we want to get rid of. But the idea is to like minimize those sorts of preservatives. Like if you have foods that can have a shelf life of 2 or 3 years, there's a lot of chemicals in there that are doing that. And I think we've just become almost lazy in our diet to rely on those things that have these very long shelf lives. Not like canning and things are different, but when it's from the preservative side, it is something that concerns me that we have a lot of things that can stay around for a very long time because of all the things we put into it.
Yeah, and I would imagine, there's a lot of tie-in here with exposomics and with what you just mentioned about preservatives with the gut microbiome. So, obviously the preservatives will affect the guts that the microbes that are supposed to be in your guts and not just the ones that are, the mold growing on the bread. But also, what I've learned, in talking to people on the podcast, for example, is the microbiome can determine whether or not a drug becomes activated or a drug gets metabolized into something inert and then doesn't work. And I would imagine that the microbiome might also come into play when we're talking about chemical exposures. Maybe a chemical that I get exposed to gets turned into something even nastier by my microbes, but it's sort of taken care of by yours. Is there anything going on with exposomics and the gut microbiome?
Yeah, so it's a very interesting linkage in there. And again, if you're thinking about your diet, you're exactly right. So you think about the foods that you eat are going to, they're absorbed through your gut, but there's all those enzymes and those microbes that can convert them. And if you think that like you have something that has a pesticide residue on it, that pesticide could also be converted. So a lot of the things that we see when we're measuring by mass spectrometry in our blood are post-microbial. They've actually been metabolized by the microbes in us. And this is like when we talk about unknowns, often the unknown, it looks like something but it's been converted by potentially a microbe, and it's just not in the database sort of thing.
Mm-hmm.
The data I've seen is clearly shown, though, is that a healthy gut microbiome actually is a very good barrier of the bad chemicals that come in through your diet. So a healthy microbiome actually does a good job of protecting the lining, breaking down things that shouldn't be in there.
So in other words, you could have two people, same exposure to the same synthetic chemical, but one of them has a gut microbiome that can basically get rid of that thing before it gets very problematic. One person might not have that microbiome, and therefore their exposure is much more dangerous.
Absolutely. Yeah, absolutely. So the idea that's like it's a very important barrier. Like so, having a high quality diet keeps your microbiome healthy and it protects things from getting into your body in the first place, like across that. So it's very much connected. So again, a lot of these things we detect are microbial metabolites in the blood. And so you can also think about a lot of the chemicals we get exposed to. They can damage the microbes that are in your gut. They can activate enzymes that are in them. So there's a lot of interactions going on with the things that come in through the exposome and the microbiome, not just in the gut, but our skin microbiome and in our lung and everything else.
Mm-hmm, mm-hmm. Just in terms of the research in general, where the technology's at, the ability of us to communicate at a distance like we are now, and just sort of the general excitement that seems to be growing around being aware of what you put into your body, whether it's food, whether it's chemical exposures, being aware of new technologies, taking more data-driven approach and a preventive medicine approach rather than a reactive medicine approach. Are you or anyone else in this field, are you guys doing studies where, you can recruit volunteers who, volunteer to give you regular blood draws and then, fill out life history forms and things like that? Is there any participatory research like that going on?
I mean, so like our group has been very much focused on the sort of classic academic sort of things, but there are many groups I'm aware of that do more of this sort of like consumer level work where they can collect blood samples and try to get that sort of information back. And so there's definitely groups that have been doing that. I think a lot of the kind of like longevity companies that have been developing, they're thinking along these lines about what we can measure. I don't think many have really gotten into this exposome space as much, but it makes sense, right? Like the idea is that these things definitely have a drive to it, but like I'm aware of some companies where they do have like kind of like super participants who are willing to give blood samples and urine samples to do this sort of work. But I think what we really need, though, are the large scale, well controlled studies, thinking about like the All of Us study in the US, UK Biobank. There's been really good studies about the combined genetic and environmental factors, but they really haven't taken advantage of those biobank samples to do this sort of analysis that we've been discussing. And I think that we need that sort of study to make the connections to say, what are these sort of actionable areas in a very rigorous scientific way to then think about like how do you go to the consumer and say, well, now we know these chemicals will cause these problems. Here's how we can help you reduce them.
Hmm.
But at this point, like I don't think we really have the full story to this point.
Hmm. And just in broad strokes here, what are some of the chemicals that people are most commonly exposed to at the highest levels that we know are or are likely to be problematic? Can you give us just a brief survey of that?
Yeah, I mean, I think that we've been hearing a lot about the perfluorinated compounds, the PFAS-related compounds. These compounds are used in so many consumer goods, and many of them are used because like they're very good at repelling water, or like that little sheet of wax paper in your pizza box. It's trying to keep the grease from getting into the box, sort of thing. So like they have these uses, but we use them in everything. And so all of these sort of PFAS and plasticizer sort of things, we are just loaded up with those sorts of compounds. And the problem is that like they don't appear to be acutely toxic in like a laboratory setting when you test it like in an animal sort of model,
Hmm.
but we don't test thousands of them at a time, which is what we're exposed to.
Right, right. We're not
So
exposed to one
yeah,
compound at a time.
and so you look at all, so you have all the different sort of to me like the consumer goods sort of space and these things we put again like how we prepare our food, like all the packaging we use in our food, those sorts of chemicals we see a lot in people's bodies. A lot of pesticides, because we use pesticides to control, weeds and roaches and all these different things. There are all these different types of chemicals and we can detect those in many different people. And then also even things like heavy metals. And so there's been a big focus on reducing lead, like in gasoline and in paint. But we still see exposures to things like cadmium and that just are in these different environments, and people have exposures that have known adverse health effects. And again, individually, you may not see an adverse effect of the level of that one thing, but if you look at them across all the things we're exposed to, I can imagine if you just looked like an overall body burden of chemicals and you had people like in a decile, you clearly know that people that have the highest levels have worse outcomes. So
Hmm.
like what are the components of it that are causing it? That's what we have to kind of start teasing apart. But until you measure it in a systematic way, it's very hard to get to that. And that's what we've been trying to build is this ability to do these large scale studies of, 10, 20, 30, 50,000 people.
Hmm. If people regularly donate blood, say, is that actually a reliable way to decrease your exposure, to get rid of things that are already inside of you?
I haven't thought about that as like a cleansing sort of system. Um, I think donating blood is good. I'd encourage people to do it. It can be life-saving for people. I'm not sure that that would really have a major effect on uh, the chemicals in your body. Just in thinking about when you're exchanging your blood, you're also probably getting rid of some good things. So
Hmm.
like it's like the net good and bad. I'm not sure if that sort of process would be beneficial. If a person had like a known exposure, like if there was some sort of accident or something, there was a big exposure, there might be a situation where like a chelation type therapy or pheresis sort of therapy might want to be done like in a very methodical way. But I don't think that donating blood on a regular basis is as effective as reducing the intake of the exposures.
And on the intake side, what are sort of the simplest ways that the everyday person can reduce their exposure to the things that are most likely they're going to be exposed to? Is that where we think about things like water filters and so forth? What are some of the major things here?
Yeah, so I mean, I think we learned a lot about these things during COVID and thinking about air quality. You can kind of go back to that time. Like if you look at the situations where our exposures to viruses were higher, you also have higher exposures to all the particulate matter and other organics that are in the air. So you may have seen those Corsi Rosenthal filters that people made during the pandemic, where you took a box fan and like those MERV filters, like for $20 you could build these things. Is air filtration works really well, and our indoor environment is usually worse than the outdoor environment because we rebreathe everything and all these things are accumulating. So to me, I think that having good airflow in a building is good. Bringing in external air is very positive. And if you can afford a HEPA filter, like I have a HEPA filter sitting in my office here, I have a HEPA filter in my bedroom in my apartment. Because if you can cleanse, whether it be viruses or pollen or these various organics, they all kind of come out in this sort of fit system. And then also, even like the things like water, is we have very good systems for, decontaminating water in our water systems. But, the apartment I live in is over 100 years old. I don't know what the pipes were made of and things. So I use a filter to filter all the water that I consume from my tap because I don't know exactly what's been coming through there. And so, the water filtration systems and the air filtration systems, to me, are very cost-effective ways of reducing those exposures. And then also, I think as we talk about the sort of ultra-processed foods, it's very clear the more processed the foods are, the more of those chemicals you're bringing in. And so reducing the types of things that are highly packaged and processed is also a very good way of reducing those sorts of exposures. And it's also just healthier for you from a dietary standpoint. So I'm thinking about the food you're bringing in, the air filtration, the water filtration. The other thing I think that people don't pay enough attention to is that when you're using chemicals in your yard or garden, the simple act of wearing of gloves, chemical resistant gloves, can protect you from those absorption exposures. A lot of organic gardeners love using rotenone. Rotenone is an incredibly dangerous mitochondrial poison. But because it's organic, people don't think it's dangerous.
What's it called?
So,
can you spell it?
Rotenone, Rotenone.
Okay.
And there's many studies that show that rotenone exposure increases the incidence of Parkinson's disease.
And this is a natural organic compound, so people think it's probably okay.
Yeah, exactly. Yeah, and so a lot of these natural compounds and things that people use in organic gardening actually can be dangerous. But it's one of those things that when you're using, like say, for example, you're spraying ants or something in your apartment, is if you're going to spray those things, you should open your windows to let those volatiles go out. But wearing a pair of gloves when you're doing the spraying is also a good idea because you want to reduce the exposures to those sorts of chemicals in that acute setting.
When when I think about chemicals and toxins and xenobiotics, the organ that I think of is the liver because that's sort of our natural organ that's focused on handling those things. So with that in mind, so first a general question. So when we think about exogenous chemicals, synthetic pesticides and plasticizers, and you name it, are these things are they usually like generally toxic? Like they get into the body and they're just going to kind of mess with stuff wherever they go? Or are there any chemicals we get exposed to that have more organ specific effects on the liver or on the brain or on some other organ?
Yeah, so there's two basic toxicological concepts there. So the dose makes the poison is something that's been used for hundreds of years. And so it's something where the more of these compounds you're exposed to, like the bigger the effect is. But yeah, there are many different compounds that have very selective toxicity to different organs. There's some things that very much target only regions of the kidney, and they're very acutely nephrotoxic. And there's others that cause problems very specifically in areas of the liver. And so it might be because of the enzymatic systems that are in those parts. The kidney is a great example because we concentrate things in our kidneys. So the
Mm.
concentrations the kidneys see in certain regions can be like 1,000-fold higher than what's in your blood because it's concentrating your urine.
I see. So if the dose makes the poison, a low dose might be seen by your other organ systems, but then a high and poisonous dose is then concentrated within the kidneys.
Yeah, exactly.
Hmm.
And so we see this throughout different systems is that all of our organs have different features, different transporters. And so they may often it's like they're accumulating it more, or again, they have an enzyme that activates it more in that region. And so you only see it in that organ. So it is something that's why we study in the sort of organ system level, because there's some compounds that just have that preferential toxicity in different
Mhm,
areas.
mhm. Um, Another general question. So, when I just think about something like the liver and I think about aging, things start to break down over time. Does there come a point in liver health as we age where the liver just becomes so dysfunctional that you're then more open to be harmed by all of these chemicals because now the liver's not going to metabolize them as it once did?
Like I don't know much about the aging of liver, but using the example of like cirrhosis, like where we know for, either whether it be alcoholic or other conditions, is that many of those conditions, and even like fatty liver, which like just comes from like over obesity and things, is that they tend to be progressive. And so
Mhm.
as people get older, those diseases are progressing. And absolutely, as people progress in either cirrhosis or fatty liver, the function of the liver starts decreasing because the systems are not working as well. And so it does increase, like it decreases the ability to break down things that need to be broken down. And so it definitely does, at least in those situations. Like as does aging alone, I just don't know the answer to that, whether
Mhm.
aging alone affects liver function in that way. I know, for example, like in the kidneys, again, as people get older, they tend to have more problems with their kidneys, but it's really a matter of getting to the point where you lose enough function. Like you can donate one of your kidneys to somebody and 50% function works really well. But it's when you get down to only have about 20% function of your kidneys that you really start seeing problems.
Mhm.
So I think the liver has quite a bit of reserve. And I think outside of those sort of cirrhotic conditions, that there's no reason that someone who's 80-year-old can't have a healthy liver.
Mhm. When we think about exposure to exogenous chemicals, it's natural to think about things that we don't want to be exposed to, that we're inadvertently exposed to. Like, there's lots of chemicals in the environment and I don't want to be exposed to the pesticides, but as a natural consequence of eating the food or doing my gardening, as you mentioned, I'm going to get exposed to these things inadvertently, but I'm not trying to put these things in my body. There's also a lot of things, a lot of exogenous chemicals that we do put into our body on purpose. um, on the subject of neurodegeneration, are there any chemicals, such as, say, pharmaceuticals, that people put into their bodies long term on purpose that are tied to neurodegeneration?
So again, like I'm thinking about, there are a few things in there. So there is good evidence for some conditions where people that take long-term anti-inflammatories, it actually protects them from those conditions.
Mm-hmm.
Knowing that inflammation is critical in many diseases. Uh, I think this is like where it's hard to kind of point to those single molecules, but if we've heard of like anti-inflammatory diets. Well, the opposite of that is someone who has a pro-inflammatory diet. So if you're bringing things in that cause, that activate those cascades, those are things that could drive those sorts of processes forward, that could cause more of those problems. We mentioned the sort of amphetamine exposures and things
Mm,
like that with like
yeah.
medications. This is like one of the challenges is that we take many different medications that have all these great benefits to us, but all medications have side effects. And I think this is something that's been understudied in that we tend to study the medications when they're going through approval. The drug company studies that drug, but 20% of Americans take 5 or more drugs. And we don't study those drug interactions over time in people. And I think that a lot of the adverse health outcomes we see are these sort of accumulated side effects of many of these different
Yeah.
medications.
So we've got drugs that we use in combination, and we don't test those combinations. We also have drugs that we use for long periods of time in combination sometimes, and we don't test for that either, just because that's not the nature of the system that does the testing.
Right. And so this was a project that we started a few months ago with funding from ARPA-H, which is this new health agency. Um, and it's designed to look at individualized pharmacology. So what we're trying to do is use our mass spectrometry to measure all the environmental chemicals people are exposed to, all their endogenous biology, and then all the drugs and drug metabolites that are in them.
Hmm.
To see if we can start looking at how these sorts of interactions of the environmental factors, the dietary factors, and the drugs they're taking, how those interactions are occurring. I think this is a place where, like, you've probably seen like a warning label where you shouldn't drink grapefruit juice when you're taking certain medications.
Yes.
That's because these furans in the grapefruit juice inhibit one of the cytochrome P450s, and so then you don't metabolize the drugs correctly.
Mm-hmm.
And like I tell people is that it's not just grapefruit juice. There's so many other things in our environment that activate or inhibit different enzymes that process medications. Yeah. And so when we think like, why doesn't someone respond well to a medication? It might be one of these environmental exposures that's interfering with that. And so, but we historically, we haven't systematically studied all these things at once, but these new mass spectrometry techniques let us do that. And so that's exactly what we're trying to do, is scale up so we can measure as many of these environmental factors and the drugs and the drug metabolites at the same time, and look at the disease outcomes and progression to see if we're seeing, explaining why some people don't respond as well to a drug.
Yeah. And when we think about chemical exposures and exogenous chemicals, it's easy to bucket things in your mind in terms of like natural versus synthetic, but it's actually not that simple. A lot of people just think, okay, synthetic bad, natural good, but you've actually mentioned several examples over our conversation of sort of all four quadrants here, where you've got natural, synthetic, good, and bad. Like you said, grapefruit and many other natural foods have compounds that are totally natural and organic that affect liver metabolism in different ways, sometimes for good, sometimes for bad. There are, you mentioned pesticides. When we think of pesticides, we think of synthetic man-made pesticides, but, caffeine and nicotine are essentially natural pesticides that you actually mentioned can have beneficial effects with respect to Parkinson's. So is there anything worth unpacking there for people in terms of good, bad, natural, synthetic, and all that?
Yeah, I mean, I think it is something where the source of it is something that's very critical, is that if you have an infection, taking a synthetic antibiotic is a good thing because it helps fix that condition. And we know that there's a lot of natural things that have that. So you have those different quadrants. And I think it's really about how you balance out those various exposures and, again, minimize your exposure to the things that we know are bad. Like, that's what it really comes down to, is how to have that balance there. I think that trying to think of some other examples here of these things is that, we have a lot of the different things, like when we've talked about these different examples of, I tend to think not so much about the synthetic versus natural, just because I think about all the venoms and toxins that nature makes
right.
Can be very bad. And the idea that many of the chemicals that we've produced have these very strong benefits. And so when we think about things like DDT, like, Silent Spring, and like we needed to ban all these compounds. But if you're in a place where malaria is endemic, the malaria is the greater threat. And so it's about balancing what things we actually need. And so if you have, if you have West Nile virus that's killing people, you might be able to handle some pesticide exposure to get rid of that. But if you don't have that, you shouldn't be using those toxic compounds. And so it's like you're trying to get this overall holistic risk assessment there. And I think that's where we tend to overuse many things that we shouldn't be overusing. So the idea is like, you want to be using interventions to control the problem, and the minimal amount you need to do that. And whether or not it's synthetic or natural isn't really the question. It's really the value it brings. Like, is it valuable to use that at that time?
Yeah.
And if it is, use it. And when it's not valuable, don't use it.
Yeah, I think an important principle that's lurking beneath what you just told us is that pretty much everything in biology is context dependent. There's rarely something that's universally good, bad, pretty, ugly. It's context is often a critical factor in terms of how something affects us.
Right. Like many chemotherapeutics are very dangerous compounds, but if you have cancer, they're good. Like you want them. And you're willing to accept some toxicity to your kidney or liver if you're going through a treatment of chemotherapy because you're trying to deal with the cancer. And so it's like always in that context of like why these things are occurring. And it's something where you have to be able to balance those risks between what is the benefit of that compound in that certain context.
There is a fair amount of genetic variability in terms of, just like variation in things like liver enzymes that metabolize different foods among humans. Is there any variability in people's ability to detox from certain environmental chemicals? Are some people better able to metabolize certain chemicals into something innocuous than other people?
Yeah, I mean, it really works the same way, whether it be the variation in the response to a medication or pesticide. There's great individual variability there. We think about like using pharmacogenomics to use people's genome to figure out how drugs work. I think there's room for this pharmacoexposomics space too to think how their exposures affect those drugs. But it also comes down to these other exposures. And this is like where I think these approaches we're using exposomics, where we're measuring the things in the blood, is what we're measuring by measuring these small molecules. It's downstream from people's genome and the proteome. And so we capture that. So if someone's not as good at metabolizing a certain compound, we're seeing the evidence of that. We're seeing the accumulation of things. Like, why does this person have so much more accumulated than the next one?
Mm-hmm.
Like, we're measuring those things in their body. And so that's like why, the idea of being able to measure things in people, whether it be in blood or potentially saliva or interstitial fluid or something, it's important to do so many of these things at the individual level. Now, if we know, for example, that there's some class of compounds that's bad for the environment, we want to use policies to reduce their use. But people are different, and we have to address that variability. But when you're measuring samples, just like a doctor takes blood out for your clinical chemistry, the exposome needs to be done at that individual level to really address that variability.
We've already covered quite a bit of ground. This has been fascinating, and obviously, this is all a work in progress. Like this is a pretty nascent area that you're a part of, and so there's a lot to look forward to in the near future. Can you give us a sense for some of the things you're working on now, some of the things that we'll start to get answers to in the next few years that you're excited about in the field?
Yeah, I mean, so there's one is that, we've talked about the exposome in a pretty clean definition standpoint, but there has been quite a bit of discussion and debate about what it is and what it includes. And so just over a year ago, several colleagues of mine, we convened a meeting at Cold Spring Harbor Laboratories in this Banbury conference about integrating exposomics into the biomedical enterprise. And one of the big goals was to come to a clean definition of the exposome. And so we've worked on this, and we have a paper coming out in Science in April that has this definition. And our hope is that like NIH and other research institutes adopt this as a clear definition that everyone uses, because it's important to kind of be on the same terms of these things. We also are working with NIH on this coordinating center for exposomics. And one of the goals here is really to kind of develop the global collaboration. Like we saw this with genetics. Mm-hmm. Until you really got everyone kind of working together on common methods and tools that you could harmonize across different laboratories, it was hard to really make some of these big breakthroughs. And so we've been working with people across the world really to kind of get to these harmonized methods so that we can bring the data from different countries and different systems into some sort of uniform system. And I think this is where you mentioned some of these AI approaches. Like we can think about like large language models and many things we've seen advance in AI, but we can use those same approaches to pull out data from the mass spec too. And I think this is where these sorts of AI approaches, when we see all these, it's very good at detecting patterns, right? When we're measuring hundreds of thousands of peaks, when we can get data on 100,000 people and hundreds of thousands of peaks from their blood, and then use that geospatial data, like I really think that's when we're going to start pulling these things together. And like these studies are like now underway. Like we're doing these on thousands of people. My big goal is I want to do Exposomics on the UK Biobank, on the NIH, all of us. But to do that many samples, like our throughput has to get faster, it has to get cheaper. And so I think that the use of AI and robotics and things is going to get us there. But doing genome-wide association studies took all the sequencers to get really fast.
Yeah. Before you can do the big data stuff, you have to create the big data, which requires you to have technology that's cheap enough to do at scale.
And so I think this is something where we're seeing more of this. We have a meeting planned in May. Johns Hopkins is a co-sponsor. It's a Exposome Moonshot Forum where we bring together people from across the world in different industries to say, how do we do that? How do we scale these things up? How do we get the industry partners involved in making the instruments better and the informatics faster, and again, getting these data systems together? So I think we're at this point where it's really trying to get more people together on tackling this challenge. But because many of the tools are at this point, we can now do this. I think that the next few years for Exposomics is really going to be an explosion because we know how much the genome can tell us about certain conditions, and we know what it can't tell us. And that's Exposomics is gonna fill in that gap over the next few years.
All right. Well, I think that's a great spot to end it. Professor Gary Miller, thank you for your time. Fascinating stuff.
Okay, thank you.
Visit mindandmatter.substack.com to find all of my content. Hey everyone, I want to take just a minute to tell you about a really cool health monitoring device I've been using called Lumen. It's a handheld, pocket-sized device with a sleek design. The Lumen device measures CO2 levels in your breath, which allows their technology to determine the extent to which your body is burning fats versus carbohydrates. Lumen helps improve your metabolic flexibility, your body's efficiency in shifting between using fats and carbs for energy. I use this device in the morning or before bed or after meals or after workouts to track my metabolism. With just a couple weeks of use, I learned a lot about which foods were causing my body to burn mostly fat, mostly carbs, or both, as well as how long I needed to fast and how hard I needed to exercise to promote fat burning. Being able to track whether your body is burning fat versus carbs will help you figure out how your body responds to your lifestyle and whether or not your weight loss and fitness strategies are actually working. Lumen is great for anyone looking to optimize their health for either weight loss or athletic performance. Their easy-to-use app allows you to track your results together with what you're eating and how you're exercising, and it syncs with other devices like the Apple Watch. Click the link in the episode description to learn more and use the code MINDMATTER, MINDMATTER, to get 10% off your Lumen device today. That's on top of the site-wide sale they are currently running, and the Lumen device is eligible to be paid for with FSA and HSA health accounts. Check out the link in the episode description for more info.